system to keep that macroarthropod and the predatory nematodes in control. And then what's the top of this food web with us as human beings? Just that we've been falling down on our job. We've been messing up because we're arrogant. We think we know better than Mother Nature. Poor little Mother Nature. You got to help her out. Yeah. Instead, not understanding what we're doing, we've been destroying the system, which means you've got to work really hard in order to grow your plants, and we're not making quality plants. So need to get this system back into the soil so that nutrient cycling can happen. We don't have to worry about diseases and pests at all whatsoever if we get this food web established around the root system and on the above ground part of your plant. The same kind of protection is going to be happening above ground as below. When a disease-causing spore floats along, lands on the root system or lands on your above ground, your leaves, your flowers, your seed, there is a layer of protective organism that a spore is never going to germinate because it never gets the food it needs in order to germinate. If perchance it did germinate somehow, it's not going to be able to get its disease-causing member to move through, it can't get through that protective wall. Something's going to compete with it. Something is going to use its enzymes to chew it up. Or we've got a predator that's going to come along and eat that disease-causing organism long before it ever gets even close to the tissues of your plant. So if you have problems with diseases, if you've got insect pests, Mother Nature is trying to tell you, you don't have the protective layer. You don't have the nutrients in this plant to allow it to grow in a normal fashion. So if you want flavor and taste, you better get this biology back into the soil. So suppressing diseases, retaining nutrients, keeping them in the soil, nothing leaches anymore. We don't have runoff, we don't have erosion. The nutrients are converted at a rate that the plant requires. Let the plant be in control of its own life. These are decomposed toxins. So if your soil goes anaerobic, if compaction happens, we need to get the biology back into that compaction zone to rebuild that structure and prevent oxygen from being excluded from some part of your soil. If you've got compaction, if you've got anaerobic conditions in your soil, then it's not soil anymore because you've lost the organisms and probably the organic matter that you need. So we've got to deal with those toxins that have been produced under anaerobic conditions. And that's another major function of these aerobic organisms, is to build structure. The bacteria make massive amounts of glues around their bodies, which means they're going to glue themselves to surfaces, grab those surfaces, hold them, and keep them in place, building that structure around that bacterium so it's protected from things that want to eat it. Come along with their strands and pull micro aggregates together into macro aggregates. And as these things are pulled together, think about the space that's left. Now we have airways and hallways and tunnels and passages that allow water to move normally through your soil. So infiltration occurs. You fill up the pores about 50% so that you've got plenty of water stored in that soil. Organic matter chewed, the dead plant materials chewed on by the bacteria and fungi, the decomposers, glued together, held in place by the bacterial glues, the fungal strands, hold water against the flow of gravity so that when your roots grow down into the soil, and of course the roots have to have space. They have to have the airways, the passageways, the hallways, the tunnels to allow those roots to grow easily and deeply into your soil. Now, a lot of people think that our crop plants only put their root systems down a couple of inches and that's it. And then those roots grow sideways. And when the roots grow sideways, they start combating neighbors' roots. This is not normal. This is not natural. Those root systems should be going straight down. And so make certain you have no compaction in your soil. What's the easiest way to tell if you've got compaction or not? You push a metal rod through your soil. Pounds of pressure, that's a compaction layer that most roots of most plants cannot push their way through. And if those roots do try to push their way through, they're going to be met by very toxic chemicals that are only produced under anaerobic conditions. There's a whole herd of organic acid. Drop the soil pH very low, drop the soil pH less than 5.5. That's a message from Mother Nature that there's something terribly wrong. This is not soil, it's dirt. Fix the dirt. Get the biology back into the soil to build that structure so your roots can go down as deep as they possibly can go. How deep does cannabis go? How far down can we find the root systems of the cannabis in the first growing season? Very easily, those root systems can be going down 10, 12 feet, not inches, feet. And if those root systems are down that deep, you don't have to irrigate because those root systems are finding the water that was stored in that soil back at snowmelt or back with the spring rains. If you've got a compaction layer in your soil, your water can't infiltrate. It can't go down and be stored in your soil where your root systems are going to be able to access it. Compaction layer means that when the water hits that compaction layer, that water's now going to run downhill and not be stored. And it's going to pull a lot of your soil with it as it goes. So now your roots can't grow deep. They can't get the summer water. You're going to have to try to figure out how much water does your plant require every day. And it's going to cost you money. So we need to get this biology back into the soil. Be able to stop irrigating, stop spending your money on water, stop spending your money on inorganic fertilizers and pesticides and weedicides. Because if we get the balances of these organisms proper into your soil, weeds don't grow. Weeds require strictly nitrate to promote their growth. They only take up nitrate, maybe nitrate, and a small amount of ammonium. What we have to do is get that ammonium component back into the soil. And so when we're dealing with cannabis, we want around just about the same amount of nitrate in the soil as ammonium. And then weeds are going to be outcompeted by your crop plant. Your crop plant's going to be getting all the nutrients it needs. Not the weeds are not winning anymore. So your crop is the healthy organism in the system. So I'd like to quickly go through what the morphology, I may be running over. Let me know when I'm supposed to stop.
You're good. You have another 45 minutes. But
another 45 minutes.
Yeah, we'll
Yay!
get some Q&A in there.
We can do this.
Okay.
So let's go over what these organisms look like. So when I'm talking about bacteria, if you took a little bit of your soil, let's say you take a teaspoon of your soil, and now you're going to mix 4 teaspoons worth of water in with that soil, and you're going to shake that so you break up all the water-stable aggregates. And you took a drop of that mix of your soil, or what you think is your soil. Maybe it's your dirt. Very rapid. So you're going to mix a teaspoon of your soil with 4 teaspoons worth of water. Please make sure it's clean water. Make sure that it doesn't have chlorine or chloramine in it. Because guess what we put chlorine and chloramine in the water to do? Yeah, to kill the organisms that would grow on the pipes on the way to your house. So that's what the chlorine and chloramine is in your water for. You've got to neutralize that before you add it to soil, before you put that water into a place that you want to have microorganisms growing. So make certain that it's not chlorinated and it doesn't have chloramine in it. In some of the other classes, we go through how you do that very simply, easily. You can make your own neutralizer. You don't have to buy it from the store. It's called compost. So let's see, where was I? So you do that dilution. Now you're going to take 1 drop. So you get an eye drop dropper, you cut one drop on the slide, put a cover slip on the slide, and then use a shadowing microscope. If you need to know where to order a shadowing microscope, go to our website. All of the information is up on the website. And we have classes, online classes that you can take, teach you how to use that microscope. It usually takes us about half a day to teach people how to use the microscope. And then we go through pictures of these organisms with you so you can identify them for yourself. It's best if you start doing this microscope work. This is the measurement that tells you whether you have healthy soil or not. So easy, simple to do these assays, so simple to do it yourself. And then you're not sending something off to a laboratory and wondering if the US Mail or UPS or FedEx have managed to forget where the address is that you were sending it to. Take a week or two for the sample to arrive if you learn how to use the microscope yourself, because you know what bacteria look like. So let's go into this. This picture up at the top, it says, bacteria, aggregates, roots, ciliate. So these things are in this picture. Before we get started here, before I tell you what these are in this picture, can you pick these things out? Do you already have enough of a background that you can be doing some of this identification of these organisms? So if this was your first field of view that you looked at with your sample, should you be patting yourself on the back because we've got some really good organisms in here? Or should your heart be sinking down into your boot? Oh, no. Oh, this is bad news. So where are the bacteria in this? These kinds of preparations, you would have to go to a much higher magnification. What's the magnification on this? Over in the bottom right-hand side, you can see this is 400x total magnification. In reality, these organisms are 400 times smaller. So yes, it takes a microscope to see them. So those are cocci, the little round back. And if Leighton, if you wanted to go and point to these as I'm talking about this, if you point out and just point to where I'm talking, that might help people in the room. So that little aggregate that's over midfield on the left-hand side, halfway between the edge of the slide and the middle of the slide, that's a micro aggregate. Now you can see some rod-shaped strands in there, very short, very narrow, round, or rods. When we're looking at these different bacteria, notice that we basically just have two kinds of morphology. There's the big round bacteria, and there are the rod-shaped bacteria. And when you're only seeing two kinds of bacteria, your heart should kind of be going, oh no, things are not good in here. In the past, this has been good because we've got that micro aggregate. There's other big macro aggregates over on the left-hand side and over on the right-hand side. So in the past, things have been good, but there's something out of whack in here now because we don't have the diversity of the bacteria that we should be seeing. We'll see other pictures here in a few minutes, and you'll be able to see what I mean. This is bad news in here. Now, that big strand that's going across the bottom half of the field, that's a root. You can see lots of different cells within that root, so multicellular. You know that that's not a fungus. Fungi are single strands. But most root systems, the root itself ought to be uniform diameter all the way along, except down at the tip. So as you look along that root, things are going pretty well. If we start in the lower left-hand side and you go along, it's pretty well uniform diameter all the way along that until, see the big divot that's been taken out of the side of the root? What's caused that? That is not healthy. That is a very bad sign. It could have been a microarthropod taking a bite, so a disease-causing or a bad guy microarthropod that comes along and eats roots. But look at that real dark black blotch right at the bottom of that bite that's been taken out of that root system. That's a fungal disease. We're looking at some fusarium, and that is a disease-causing fungus that, if left unchecked, is going to take out your plant. So probably if we look at other roots from this particular plant, we would see more signs of attack. But that tells us that this root system no longer has that protective bacterial and fungal castle wall around it. It's naked, it's hanging out there, and it's trying not to be noticed by diseases and pests and problem organisms, but it's been found. If you have this occurring on your plant, you want to, as quickly as possible, inject into the soil the proper biology around these root systems. You can get the competitors, the inhibitors, and the consumers back into the soil, and you can repair this damage if you're fast enough about it. If you are waiting for the above ground part of the plant to tell you something very bad is going on in the root system, your plant is dead before you know what's going on. So you want to be using this microscope as a way to tell you about the health in the soil. Do you have the organisms or not? And so some of these kinds of things I'm pointing out, you want to be aware of, so you can see the messages that nature is trying to send you, and you can get the message soon enough that you can do something about it. So in the past, things have been healthy, but right now we've got two indicators of uh-oh. And then, of course, the third indicator in here is the ciliate. So that gray, big, kind of oblong, grayish structure, that's a ciliate. If we were watching a vet, ciliate would be moving around. It would be zooming and eating bacteria, moving very rapidly on those little cilia that are coming out of its body. So that characteristic that all ciliates have to have cilia on part or all of the body. You can see inside the ciliate all the bacteria that it has consumed. So a ciliate eats about 10,000 bacteria a day. It can't stay alive without that massive amount of bacteria, but the bacteria are typically growing very, very well. In soil, lots of foods, lots of organic matter, especially where you're around a root system. So that ciliate only occurs, it only can be competitive when oxygen has dropped below 6 parts per million and you are now in a reduced oxygen and anaerobic environment. Well, anaerobic means that fusarium that will build structure, that will attack and compete with and consume and inhibit these bad guys. Your soil isn't soil, it's dirt. So get the biology back into that system. So moving on, and I'm assuming that if there's any, glaring questions that people just, answer, that Joshua or Leighton or Steve would call on them and get their question answered. If not, I will continue. So this picture, we've got fungi in here. Can you see those strands of fungi? What are the criteria for beneficial fungi versus bad guy fungi? Most of the good guys, all of the good guys in this picture, cannot grow in plate cultures. We have no knowledge of how to grow these beneficial fungi in laboratory media. So we've missed them. When people rely on plate counts or when they rely on respiration to measure fungi in the soil, they're missing 99.999999999% of the fungi that are actually present in the soil. So you have to use a microscope or you have to do DNA analysis. With your microscope, it's, those microscopes cost about $300. Don't buy the camera on those microscopes from the microscope company. They are worthless. Use your cell phone. much higher resolution, much easier to use. And you're going to be buying a cell phone anyway, your smartphones. Um, so why invest in another camera when you've got a better one? So just hold your cell phone up to your eyepieces. It'll take a little bit of steady hand, or maybe you buy a holder so that you can always put your cell phone exactly into place and take a picture of what you're looking at. If you don't know what you're looking at, you send the picture to us and we'll identify it for you. We do ask that you take the microscope glass first, though, because you're with this talk, you're only getting a very small amount. I mean, out the characteristics, so you could at least get a good idea of whether it's a fungus or something else that you're looking at. So looking at this fungus, so taking the one strand that begins just close to the left-hand side, upper left-hand side, and looking at that length, fungal hyphae to be uniform diameter all the way along. So that strand is uniform. Now you get to that right-hand side of the picture, see how that strand is now out of focus? You're going to have to use your fine focus on your microscope and focus along on the rest of that fungal hypha to make that decision that it is uniform diameter all the way along. Notice the branch that's coming off of that strand in that upper left-hand side, and notice that the diameter of the branch is different from the first strand that we look at, and that's fine as long as along the strand it's uniform diameter all along that strand. So then we know that that's really truly a fungus. Well, the next thing we want to look at is it colored or not? And it doesn't really matter which color, as long as it's a different color than the background. So look at the background in this picture. Look at the color of the fungal hyphae, and yes, this is a colored fungus. So most likely a good guy. When we're dealing with bad guys, they are clear, they're colorless, they're transparent. So one of the characteristics that we're going to use to rank the fungi in your sample, determine whether they're most likely good or they're most likely bad. So these nice colored fungal hyphae point in their favor. They're probably good guys. Now we want to look at the width of this strand. Beneficial fungal hyphae are typically 3 micrometers or wider. There's some exceptions, but they're rare. So, most of the time you're going to be making the correct assessment. How do you determine the diameter of this fungal hypha? Well, there's a built-in scale for you, because we know that the smallest bacteria have a diameter of 1 micrometer. They're usually cocci, they're usually little round guys. So where in this picture can you find a single small bacteria? You look at most of this clear grainy material, all those little grainy bits, those are bacteria, but you can't pick them out. These aggregates are really well formed. There's lots of them, so this is a really nice, well-structured compost. Yeah, this is really good looking. But where's the bacterium that we can find to use as our scale? Look in the upper right-hand corner, and you can see a lot of little individual bacteria. Look at how many different sizes and shapes there are. There's short, fat bacteria. There's there's really small round bacteria. There's medium-sized round bacteria. There's some larger round bacteria, and just lots of different sizes and shape. Yes, good diversity. That's what we want to see. So far, I am really happy with this sample because things are looking really good. So find your smallest bacterium up in that right-hand corner. And now, how many of those bacteria that size, how many of them can you line up across that fungal hypha? And so just think about, the 1 little dot, 2 little dots, 3 little dots, 3.5. We have a fungal hypha of 3.5, maybe 4. Close enough. So, do we have another point in favor of this fungus being a beneficial fungus? Yeah, it's wider than 3 micrometers. So we got 2 pluses, no minuses. Now, we know that fungi that have these little crystalline structures on the outside of the hypha, as you look along that fungal hypha, you can see little dots, little crystalline structures on the outside of that fungal hypha are crystals. An oxalate crystal is something that the fungus uses to hold and bind positively and negatively charged molecules and keep them right there on the outside of the fungus in storage for when the fungus is going to need that. So if you see those little crystalline structures on the outside of that fungal hypha, that's another sign of a very beneficial fungus. So now we got 3 positives. The fourth thing that we're going to look for are crosswells, septa. And as you look along the length of that hypha, there are no septa. There are no crosswells. There's nothing that closes this cell off from the next cell. So it's not going to be the absolute best beneficial fungus, but all the other indicators are, yes, these are good guy fungi. So you can see other strands of the same fungus. You have to remember to focus. The only part that you can actually say is a particular diameter is the part that's in focus. Real sharp edges, clear edges. Otherwise, out of focus. And so, you've got to follow the fungal hypha along with using the fine focus. Now, are there any other? There's a little tiny light brown strand. Can you find that one? So that little tiny bit, look at the fact that it's narrower diameter than the good guy that we've been looking at. But the diameter on that little bit, that smidge of fungal hypha in the corner, it's about a 3. And so it's still a good guy fungus. It is colored. It's a lighter tan color. It's not the dark. But it's still colored. It's still good diameter. Can't really tell whether it's got oxalate crystals or crosswalls, but probably a good guy. So let's keep going. Right almost center field, a little bit down from center field, there's a fungal strand that's quite wide diameter, but it's clear. It's colorless. So the clear, colorless characteristic is like, that's not so good. But the wide diameter, because this guy is up around 5 micrometers diameter, that says, yes, it's good. So it's probably kind of a neutral. It's probably not a disease causer. It's probably not the best. It's not going to be really helping our plant all that much, but it's not going to be hurting. So, okay, we've got fungal biomass. That's great. Can anyone disease-causing fungus in this picture? It's, if you look almost center field, but go up a little bit tiny to the left, there is a very narrow, clear strand of fungal hypha. It's hard for me to get any more descriptive, so hopefully Layton or Joshua is pointing it out for you. It's a little out of focus, so it's a little hard to see, but that particular strand of fungal hypha is only 2 micrometers. Clear, it's colorless. There are no crystalline structures on it. This is a disease-causing fungus. The particular kind of disease-causing fungus here is Pythium. And how do I know that? Because if you take some of this material, anything that grows on plates with that medium. So can I scare the living daylights out of you by showing you a plate? Look at what happened. This is what's in your soil. Oh no, you're going to have a horrible disease problem in this field. You're going to lose your crop if you don't go out there and you got to buy this particular fungicide from me so that you don't lose your crop. And so you spray the fungicide because, you're terrified you're going to lose your crop. This guy is telling you that here's proof. What does the fungicide kill? Doesn't kill just the Pythium. It kills all your good guys as well. So you've lost the beneficial organisms that would compete with, inhibit, maybe actually even consume those disease-causing fungi, and the only thing left in that is the pith. So you have a massive outbreak of that fungal disease, and see it as a self-fulfilling prophecy. This guy told you, oh no, you got to do something to destroy the disease causer, and instead has set you up for the disease taking you out. But then forever, you will now have to be using those toxic chemicals to try to control that problem, because you've destroyed the very thing that Mother Nature has in that soil to compete with, inhibit, and consume the problem. So we can't be falling into that trap of giving our money away to people who want to sell you something you absolutely don't need. Maintain and improve the beneficial organisms in your soil, and you can just laugh at people who say, oh my God, you've got pythium in your soil. We've grown plants in this material for years, and we can show you the pythium. It's present, it's there, but it cannot compete. It cannot win under aerobic conditions. When there are lots of beneficial fungi around, that thing will never grow. It will never cause disease. Kill your beneficials, oh yeah, then you got a problem. Okay, so let's keep going. Is this a good fungus or not? Yeah, and you can even see cross walls in this particular picture. So nice, dark, it's almost a blue color in here. And so we're looking at foods into this material so that you get the fungi doing all of their jobs. Okay, this is a root system of an onion. It's a microscope that you buy. You just buy attachment, cost $1,000 for the attachment. So you have to decide whether it's important enough for you to be able to measure mycorrhizal colonization in your root systems. So you get the attachment. Please get epifluorescent microscope attachment with the halogen bulb. Don't get the mercury vapor lamp. Okay, and if you're wondering why, you and I can discuss these things later on. Wanna just rinse your root in a little bit of acidified water, lay the root on the slide, and scan along the surface. So the magnification here is about 40x total mag, and you can see all of the parts of the root system colonized, active exchange sites. With the plant, you can see the amount of the plant that is not colonized. It's just that simple. You don't have to be spending $50 per sample to send it off to somebody that has to do quite complex water. And the fungus says, okay, no problem. Because I already have to collect those things for myself, because the fungus only gets energy from the plant. It only gets sugars from the plant. That's what's being exchanged. So the fungus is going to go out and find nitrogen, bring it back, going to find that phosphorus, bring it back, going to bring some water back with it. And at the arbuscule site, it says, okay, plant, here you go. Here's your nitrogen, phosphorus, water. Now I would like to be paid. So the plant says, here's your sugars. And then the plant says, now I need some cobalt and some calcium and some, I don't know, strontium. And so the fungus says, okay, fine. Goes out and finds it. So the two organisms, the plant and the fungus, both benefit. They both grow better. Your cannabis is going to be healthier. All cannabis require mycorrhizal colonization to be healthy. So get these mycorrhizal fungi into the root systems. But please remember that this is not the only fungus, that part of the root system that it's in. It doesn't protect all of the root system. So take a look at the number of roots in this picture that don't have the mycorrhizal fungus in it. Those still need to be protected. You still need the decomposer fungi to protect that root system against diseases and pests. You still need the bacteria in here as well. So simple, easy way to look at your root systems. And a microscope with the epifluorescent attachment never loses value. You can always sell your microscope if for some reason you decide you don't want to be doing it anymore. but it is such a useful tool. I can't imagine that if you get a microscope, you'd ever decide that you didn't need it. Makes things so much easier to know exactly what's going on and exactly what to fix to prevent any further problems. Ectomycorrhizal fungi, bit different than the endo, than the arbuscular mycorrhizal fungi, because with the ectomycorrhizal fungi, the colonization is only one cell layer deep on the outside of that root system. These are pine seedlings, and the seed was planted. You can see where the seed was planted. The roots go out from that point. So the above part of the plant pulling carbohydrates and sugars through photosynthesis, feeding the fungus. When the pine seedlings are this young, um, there is more fungal biomass being fed by the by the plant than there is plant biomass. And that's really very typical of the mycorrhizal colonizations. You'll usually have more mycorrhizal biomass than root biomass because the fungus is acting as an extended root system for the plant. It's cheaper for the plant to grow the fungus than it is for the plant to grow roots. So, nutrient collection, water collection, building structure, extremely beneficial to your plants. But please remember, you still need the rest of the food web to have normal nutrient cycle, building good structure, holding water, and so forth. Fungal hyphae, when you're looking in your wood chip pile, when you're looking in a compost pile, these are the kinds of strands. These are actually hundreds of strands all growing together. And this is capable of holding all of this group of woody materials together. So if you pick up one of these little chips, you would then be able to pull all the rest of the chips that you've seen in this picture up as well. Looks like a wind chime or something held together by these fungal strands. These are good guy fungi. This is what you want to be seeing in your compost pile and in your wood chip pile. Moving on to the predators. So this is a flagellate. Flagellates eat bacteria. You can see a couple of little bacteria inside this flagellate's body. You can also see the little flagella coming out in front as well as one going behind. And so that's how the flagellates chase down the bacteria. They will move around through that soil, engulfing and consuming the bacteria. And you can see lots of bacteria in this picture. There's a diversity of different sizes and shapes of the bacteria, a little bit limited, not exactly the amount of diversity I would want to see. And look at the aggregates. There are no macro aggregates in here at all. So I know, very first field of view, we don't have fungi. Uh-oh, this is not going to be the best of conditions for your root systems to be growing in. This is going to compact very easily. So we need to get that fungal component in here. Yep, you got the bacterial part of the food web. Lots of bacteria. We've got some bacterial predators in here, but fungi. As you look through more and more and more fields in this slide that you prepared, no fungi, no fungi, no fungi. So what do you have to fix? Got to get the inoculum or the fungi back in there. So flagellates have an interesting hitch in their get along. So as they're moving through the soil, chasing those bacteria, because the flagella wrap around the outside of their body, it causes drag in one condition. So the flagellate's going to be zooming along, and it's going to move slightly sidewards, so you see that bumble. And so when you see that bumble, oh, yeah, this is a flagellate. I don't have to worry about the fact because it's moving so fast, I can't really see if it's got cilia or if it's got flagella. The bumble tells you flagellate. Okay. Nematodes. We identify nematodes based on their mouth parts. So when we're looking at this particular nematode, it just has a simple mouth. It's, kind of rectangular, wide opening so that stoma can close in a little bit. and that rectangle can become much narrower, kind of cylindrical. So this tells us that this is a bacterial feeder. When we're looking at fungal feeders, we should be seeing the spear in its mouth. So the strand that goes from upper left to lower right, that's a fungus. And the nematode that's in about middle of the field to the upper right, that nematode has used its spear, and you can see the spear inside its body. It's been pulled back a little bit because the nematode used that spear to puncture the cell wall of the fungus, and you can see the liquid being sucked into the nematode. And so the nematode's going to empty out that fungus and then move along that fungal hypha and puncture at another place and get another good juicy meal. And that's how isn't soil a nice place? All the things eating other things and the way they do it. Woo! So we're looking for that spear inside the mouth. Notice that there are no knobs. There's no little round knobby things on that spear. And so the lack of knobs tells us this has to be a fungal feeder. Moving on, here's a predatory nematode. Right about center field, you can see the big open mouth on that nematode, and one side, the upper side, has a tooth. And so you're looking for a tooth, one or more teeth in its mouth. You can see what that nematode does. It's going to go up to this root-feeding nematode, open its mouth, and stab that nematode with its tooth and pull that nematode into its mouth. And then it sits and chomps away on that nematode. Then it opens up its mouth and takes another bite, pulls in until that nematode has been completely consumed. And since this is a root feeder, it's favorite foods. Most predatory nematodes that we see are going after root-feeding nematodes. If there are no root-feeding nematodes around, then the predatory nematode will go after whatever nematode it can catch. But it is especially good at reducing the populations of root-feeding nematodes. And it's probably because root-feeding nematodes have to be chock full of really good, juicy plant tissue. So they're especially yummy to a predatory nematode. This is a real treat. Whereas, those bacterial feeders, those fungal feeders, they're eating not so wonderful food resources. So the predatory nematode is going to choose to go after the root feeder. Now, are there other things that cue a predatory nematode to eat root-feeding nematodes? Not that we're aware of. So we have lots to learn about these interactions. But a good way to deal with a root-feeding nematode problem is to add the beneficial nematodes. So the bacterial feeders, the fungal feeders, and the predatory nematodes. We typically can take an outrageously horrible infestation of root-feeding nematodes down to not detectable in the course of perhaps as short as 3 weeks, but certainly within a single growing season, the root-feeding nematodes are non-detectable. Much more effective, and this cure lasts forever until you do something stupid to kill the beneficial organisms. You have a solution to this problem that will maintain itself for as long as you care to maintain the soil. You're going to have these benefits. So get these organisms back into your soil, and you don't have to work so hard. Ooh, there's the root-feeding nematode. See the big knobs? See the spear over on the left-hand side? That big crystalline structure. It's very light refractive. And then those knobs. That's what we're looking for. The first and foremost indicator that this is a bad guy. The reason those knobs exist is that this root-feeding nematode is going to try to puncture the cell wall of the plant cell. And that cell wall is always massive. If it's a healthy plant, this root-feeding nematode is not going to be able to puncture that cell wall. If this is a plant that's stressed, that doesn't have the nutrients that it requires, easy. This root feeding nematode is going to approach that root system. It's going to, its muscles that attach on those knobs are going to pull that spear back and then push really hard, puncturing the cell wall of that plant root cell. And then the root feeding nematode sucks out the juice from that plant root cell. As soon as it's done emptying out that cell, this root feeding nematode is going to move up to the next cell, repeat exactly the same thing. And in the course of a day, this root feeding nematode can take out 10, maybe 20 root cells. And so now you've got a dead spot on your root, open to infection by any kind of disease-causing fungus or bacteria or whatever organism wants to move in and start to harm your root even more. So it is a focus of infection. So we really want to make certain that you don't have these root-feeding nematodes in your soil. And if you start to see them, you know that nature is trying to send you a story, send you an information, trying to tell you something, and you need to fix it. So what's the fix? Get the beneficial organisms back in here, the castle back up around that root system, so this root-feeding nematode cannot even detect that there's a root there. Nothing is going to come out. Nothing's going to be escaping from that root. No exudates, no information that there's a root right there. Totally and completely masked by these beneficial organisms. Make sure that you've got the predatory nematodes wandering around that root system. Well, if there aren't root-feeding nematodes, then you've got to have the other nematodes for the predators to eat, so that the predator, which is on guard, gets to the root-feeding nematodes long before the root-feeding nematodes get to your plant roots. So these organisms do all the work in the soil. They perform all the processes that we have to have, and that we were convinced that we were going to have to apply all these toxic chemicals in order that we could put these benefits into the soil. And it was a lie. We didn't know the damage that we were doing. And what really set the stage for all of that was the advent of the mechanical plow, where you can till your soil more than once a year. Because, of course, if you're involved in keeping that plowshare in the ground, making sure the animals are pulling, all of that process is very physically taxing. And so you don't till again and again and again and again. Instead of tilling, you go out and you use a hoe and you take the weeds out. But if you understand biology in the soil, the weeds won't grow. The seeds don't germinate if we get that fungal to bacterial biomass ratio back up to where it needs to be. What you need to realize is that bacteria, the glues produced by bacteria, are going to be alkaline. Aerobic bacteria produce alkaline glues. Anaerobic bacteria produce acidic conditions. But got good aerobic soil, those bacteria are going to be producing alkaline glues. Nitrifying bacteria only produce the enzymes to convert NH4 into NO3, nitrate, ammonium converted into nitrate, under those alkaline conditions. And so all of your NH4 is going to be converted into NO3 when you're in a bacterial dominated soil. Now, when you're trying to grow veggies, when you're trying to grow brassicas and cole and kale crops, when you're trying to grow microgreens and carrots and celery and things like that, you want mostly nitrate, just a little bit of ammonium. So you want that bacterial system operating. But when we get to cannabis, that needs to have a ratio of more like 0.75 units of fungi for every 1 unit of bacteria. That means a significant amount of the nitrogen, the soluble inorganic nitrogen, is going to be present as NH4. And it's that NH4 presence that is going to prevent the germination of the weed seeds. So if you see weeds coming up in your soil, then now you know exactly what's wrong. And you want to get back in there with good fungal foods or maybe a fungal inoculum so you get the balance right, so that you're not promoting the weeds, you're promoting your cannabis. What we see with cannabis is also maybe we can influence the ratio of THC to CBD. There are different kinds of CBDs, and so which kind do you want? And there's some initial work that has been done that suggests that the ratio of fungi to bacteria influences or maybe even controls the balance of those kinds of compounds in your cannabis. Now, I think some more recent data is beginning to suggest that different species, different cultivars of cannabis are going to be affected to a greater or lesser amount based on what that fungal to bacterial biomass ratio is. So we need to know cultivar by cultivar, species by species, exactly what is the proper balance of fungi to bacteria in your soil to get shifts and changes in the chemicals that are present in your buds, present in your product. So early days on some of this, very clear that in I work in strawberry, I work in other crops, that this ratio of fungi to bacteria strongly influences the flavonoid concentration. It strongly influences the terpenes and the tannins. So it's something clearly that we need to pay attention to, but a lot more research needs to be done to figure out, the different types of cannabis, exactly what the proper ratio of fungi to bacteria is to produce the product that you want. So, how far down do roots go? I touched on this just a little bit before. How do we get the roots growing down as far as they can grow? You've got to get rid of compaction. This is from a soil where we made certain to go in, and for the last time ever, we tilled the soil, we tilled the compost into that soil, so we knew we had the biology down deep into that soil, that the soil was had the right biology into it, and it's down to a depth of 4 feet. We then planted, so this is Hendrikus and I were doing this research. Hendrikus planted the seed on July 15th, and when you talk to the golf course people or you talk to agronomists, they say, oh, the root systems on this grass only goes down 4 to 6 inches, right? Because normally the compaction is at 4 to 6 inches. If you break up that compaction by tilling and making certain that you can get inoculum of the organisms back into that soil, so structure is built, when you plant the seeds and those seeds start growing, and the roots start growing down, how deep can those roots grow? So we harvested this by pushing in a big PVC pipe, pulling everything in that piece of PVC pipe up out of the soil, taking the PVC pipe off, and then rinsing that column of soil. So what you see here are the root systems. This is all roots, and this came from a single seed of that grass. Harvested on November 6th. So what is that, 3.5 months after this was planted? Dug it up and look at how far down the root systems are. Hendrikus is a 6 and a few inches tall person, and he's holding that interface of the above ground from the below ground just a little bit above his waist, and those root systems that are tucked in the ground. So this is at least 4.5 feet deep in 3 months. Would you have to water your lawn if you had root systems growing this deep? With time, this root system will get even deeper. So we have people that are bringing us evidence that root systems of ryegrass like this can grow down 25 feet.
Do you have to worry about nutrients for your plant? Do you have to worry about water in the summertime? Absolutely not. And this lawn stays beautiful, bright green through the worst droughts. So no weeds, no diseases. This is what we need to be doing with our plants, is getting those root systems down. Think about what it means for growing cannabis in a pot. We need to have much deeper pots in order to allow that cannabis to grow in a good, healthy fashion and develop the flavors and the tastes we want. This is from work that was done by the USDA back around the turn of the century, the Civilian Conservation Corps, Conservation Research Institute, which today is now the NRCS of the USDA, the Natural Resource Conservation Service of the USDA. In this project, they went out to the Great Plains of the United States and dug pits, so for each of these different plants, essentially a separate pit. So they dug down 40 feet into the ground. They then brought a fire truck out and washed that surface so they could actually see the root systems, just very similar to what we did in this kind of picture. So you could see the root systems. They then put glass plating, 40 feet deep, glass plating up against this surface, tracing paper, and traced the root systems by hand. The scale on the left-hand side, both the right and the left-hand side, goes down 25 feet. The most productive grasses are the ones that have their root systems down at 25 feet. Less productive, the root systems are only going maybe 15 feet, 10 feet. You can tell those kinds of grasses that most pasture people would consider weeds by the fact that their roots don't go down very far. So look at the third plant over from the right. See how those root systems just don't go down? That's a typical characteristic of weeds. Weeds don't invest much energy at all into their root systems. Weeds are gonna put what little roots they can, suck all the nutrients out, suck all the water out, produce seed. That's the point and purpose of a weed. These other plants have a longer term strategy. They typically have a much longer life cycle. They may be perennial, in fact. So most of these plants that have deep roots are the more beneficial. Where do you see cheatgrass in this picture? Can you pick out the most horrific weed in the western part of the United States that any pasture person, and it is crazy if that weed shows up in their pasture, because it's bad stuff. Can you find it? Look way over to the left-hand side, the very first plant on the left-hand side. That's cheatgrass, Romus pectorum. It only puts its root systems down maybe 2-3 inches into the soil. It may look beautiful green in the springtime, and you've got a wonderful pasture, but this plant put all of its effort into producing seed. And if you've ever walked through a patch of cheatgrass, you know just how nasty that seed is. It cuts you. If you've got any kind of bare skin, out, you can't walk through that area. So production of a lot of seed blows out. We have to change the conditions in that dirt and convert it back into soil. So this is probably about the last second. I'm probably just about finished with my time, right?
Q&A time.
Do I have time to finish this one talk with this one thing?
Yeah.
Okay. So we're looking at succession and what actually drives succession. Probably all of you know that, Mother Nature starts out with some horrible disturbance, a flood, landslides, a fire, where you're down to bare soil. And what grows in bare soil is going to be strictly bacteria. Photosynthetic bacteria are typically the first things that come in, so a totally bacterial-dominated system. The only thing that can start to grow in that system is weeds, weedy species that don't put a lot of effort into their root system, produce lots of seeds, but it leaves the surface of that soil gets covered. So to protect the Mother Nature wants to protect the surface of that soil so erosion is stopped, loss is going to be low. But they're not highly productive in terms of developing a food web that's going to grow the later successional species back. So, at least we're started. At least now there is some cellulose coming into the system, some fungal foods, which means now we get that fungal component. So if we look at down at the bottom of the page, under that stage of succession, you can see that we've got 100 micrograms of bacteria, but only 10 micrograms of fungi. Very bacterial dominated, but at least starting to select against some of the weedier species. So keep building that fungal complement, and sooner or later, you're going to see a shift in the plant species that can germinate and start to grow. Some of those earlier successional grasses, some of the forbs, the herbs can start to survive. Vegetables are what are going to grow in that system where we've got 500 micrograms of bacteria and 50 micrograms of fungi. Fungal bacteria ratio of 0.5. But of course, more of these plants have higher amounts of cellulose in them, fungal foods, lignans as well, that's much more a fungal food. So starting to develop more fungal biomass. When we get to cannabis, it's right at that split between the second block of soil there, the 500 to 250, and the equal biomass of fungi to bacteria, 600, 600. So some of our cultivars of cannabis grow very well, and that 0.75 to 0.8, get a lot of THC out of those particularly. Now as we keep improving that fungal biomass, other kinds of cannabis grow even better, where we have more of an equal biomass to up to where we have 2, maybe 3 times more fungal than bacterial. And what we notice is that there's a trend towards more cannabinoids being produced under those fungal dominated conditions. Typically this transition where we want from highly productive grassland row crops at 1-to-1 ratio, fungi and bacteria, that's 600 to 600, as you see down at the bottom. And as we start going to fungal dominated soils, we're starting to grow shrubs and vines. And deciduous trees, ratio of 800 micrograms of fungi to 500 micrograms of bacteria. On into the old growth forests, minimum amount, 7,000 micrograms of fungi to 700 micrograms of bacteria. The most productive ecosystems on this planet, old growth, true old growth forest, 70,000 micrograms of fungi to 700 micrograms of bacteria. 3/4 of the weight of a certain amount of your soil, 0.75% of that soil is going to be fungal tissue. So think about the USDA, and the USDA says biology doesn't occupy more than 5% of the volume of the soil. What soil are they looking at to come up with that massively low number? They are only focusing on highly disturbed, very tilled soils. You can call it soil when you're only looking at 5% of the volume of your unit of soil is biomass. No, that's dirt. Call it what it is. We've got to get the biology back into that soil. We've got to get half of the weight of a volume of your soil being fungal biomass, or at the very least being biological. So this shift from nitrate, lots of nitrogen, to almost strictly ammonium when we get to the most productive of conditions. Where does cannabis grow in this system? Right there in the middle. We want equal amounts of nitrate and ammonium, and we get rid of the weed factor to select and determine what the actual composition of your bud are going to be, of your product. So summarize this whole movement through succession, from bare soil up to conifer. But then, why isn't everything an old growth forest? If this is what nature is trying to do, why hasn't she been successful? Because disturbance. Fire, floods, tillage, volcanoes. These are all disturbances that are going to move things backwards in the process of succession, and of course nature starts to rebuild. And then another disturbance happens, and then she starts to rebuild, and another, so how many times has the soil in your agricultural fields been old growth forest? How often has it been bare soil? So trying to understand a little bit of that history of your soil over the last 10 years is really necessary. You want to have some idea of what's been happening and therefore why you've got organisms in your soil and what do you need to add to bring that soil to a really healthy condition for your plants. So, do we have time for a question and answer? I would go on to compaction if we don't have any questions.
Now let's do some questions. We got some questions about any of this right now. Yeah.
Thanks, sir. Thank you, Dr. Ingham, for your presentation. My question is, one, are you in favor of cultured mycorrhizal, fungi, or bacterial inoculants? And two, what is the most efficient way to build that castle wall around the rhizosphere before going to plant? Is that gathering old growth forest soil, making a compost tea? Is that from my compost pile? Is it a mixture thereof? Anaerobic plant fermentation? What, in your experience, is the best way to build that initial castle wall?
Yeah, interesting questions. Thank you. So the first question is, I'm more in favor of mycorrhizal inocula or microbial inocula. You first of all want to find out what you're missing in your soil. If your bacteria are already there, why enhance more bacteria? Maybe if you're lacking diversity, okay, then you would think about maybe buying something that has a bacterial inoculum in it. Just you want to be a little careful about the bacterial inocula. Do you already have them in your soil? Are these really going to be benefiting the plant you want to grow? Look at how many different species of bacteria are actually present in the bacterial inocula available on the market. Great. How many species of bacteria should there be in good, healthy soil? 75,000. And so you probably have way more than 2 bacterial species in your, even in your dirt. You probably have quite a few bacteria. So enhancing that bacterial inoculum thing is like, what are you gaining? Well, if things are really, really bad in your soil, in your dirt, then yes, a bacterial inoculum would be useful. But why not go after some really well-made compost? You want to be looking at any compost that you're considering buying, using as an inoculum. You want to take a look at it through the microscope so you know what's in there. And is it worth your time to be messing around with that material? There are a lot of soil food web advisors in the world these days, and they all make good compost. Compost containing all the biology that you would want to be inoculating back into any of your agricultural systems. It can help you shift more fungal, more bacterial. Do you need more protozoa? Do you need more nematodes? We can help with that. So find your local soil food web advisor, the consultant, and get working with them. Anybody who wants to know where their closest consultant might be, get a hold of us at soilfoodweb.com. And we maintain lists of people. We do have some of the people who have graduated from the program are on the website. We have other people that are very close to graduating, so we want to get you a hold of them. It's hard to because then they would make an inoculum of really good compost for you. You don't need much. You know, you need maybe 5 or 10 pounds of really good compost to make 500 gallons of compost tea. You're multiplying the organisms during that 24-hour tea brew. You can shift for more bacterial or more fungal, depending on the food resources that you would be putting into the system. So that's a much better inoculum. The question included, you know, should I go out to an old growth forest and take an inoculum from an old growth forest? The problem is, you could be bringing diseases and pests back with you. You go out to a forest system, yep, it's old growth forest, it should be pretty healthy, but what if you pick the one spot that a tree died last fall from fungal diseases? Yeah, this is not actually a healthy part of this old growth forest. So many of our old growth forests, for example, in California, I really don't know anywhere in California where the old growth forests are healthy. They're all under stress because of all the air contaminants, because of the way we treat any of our land. We are doing really idiotic things. We're destroying water movement, we're destroying the flow, because every once in a while we get annoyed by this stream leaving its banks and flooding some of the houses that are right along the creek. We turned the whole bottom of that creek into concrete. And now it's just a concrete culvert. Well, how do you recover water table if you've just put your creek into a concrete bunker? You're not going to have the water moving out back into your soils as it should be doing around that creek in the middle of the summertime. So it just harms all of the ecosystems all along that channel, and they're not getting water replenishment. We've got to take the concrete out of our streams and put it back into a normal condition. LA drives me crazy. I can't even drive through that city without just kind of freaking out with all the, do we realize what we're doing? Well, no, we don't. And people lie to us all the time because they want to make money because we're Captain These corners.
So, can I ask a question? Assuming I know how to use a microscope and I can identify the proper microorganisms, and I don't want to go out and talk to a soil food web consultant because maybe there's not one in my area, and I want to do this on my own and go out and collect. Can you talk about your method and collection and maybe where places are to do that and how you would think about that?
Yeah, you really want to, if you go out and collect a handful, you go into an old growth forest, brush away organic matter, and you see those nice white strands on the surface of the soil. You can take, a couple of teaspoons from that. You've got the inoculum that you need. Put that material into a plastic bag. You don't seal it all the way. You don't want those organisms to use up the oxygen in that plastic bag while you're carting that home. But once you get that back, you're going to want to add that inoculum of those really beneficial organisms, and maybe a couple of bad guys in there. You want to mix that into your compost pile at the beginning of the composting process. Because you want either the temperature that's going to be developed in a thermal pile or in a worm bin, you want that material to either pass through the digestive system of the worm or contact the outside layer of the worm. Either any one of those three things will get rid of the pathogens, the pests, the parasites, the problem organisms. The weed seeds are going to be removed. They either contact with that earthworm or thermal temperature developed in the thermal pile. If you're afraid of weed seeds in the worm bin, you're going to have to do the thermal thing first and then go through the worms. So we'll deal with all the problem organisms through that composting process. Plus, we will then enhance all of those beneficial organisms that were present in that material. They should be growing in your pile now. So once that thermal pile comes back down to ambient temperature, You can use that material to make your teas, to make your compost, make your extracts. Put the compost out. Maybe for the last time ever, you will till the compost into the soil, especially if you have compaction layers in that soil. And I don't know if the slide they're still showing, but there's a compaction layer for you. Either in a landscape or in an agricultural system. How far down is that compaction layer? Well, dig a hole and you find that compaction. And so how far down do you have to till that compost in, break up that compaction? So, then above ground, we're gonna want to be applying the compost tea to the above ground part of the plant, because in that first growing season, where you've at least started putting the good biology back into that soil, you know those are only beneficial organisms that you're putting back. And you can use your microscope to make certain. So we show you the different kinds of bad guys that you need to be paying attention for, looking through your sample with your microscope. So you've seen ciliates, you've seen what happens to root systems. So those are two, but there's a number of other indicator organisms that we train you to be able to identify. So you want to spray the above ground part of your plant with the compost tea, because growing organisms make the glue layers so they instantly stick to the bottom of your leaves, to the bottom of your buds, to the bottom of the flowers, as well as the top side. When you want to get these organisms into the soil, when your growing plant is already starting to grow, you don't want to disturb it because you're going to harm the root system, then you will inject these organisms into the root system. So you want to buy an injector system. We've tested a lot of different injectors, the actual gun, the metal rod with the pump on it to pump the tea down and squirt it out the bottom of that pipe. You have to look out for the pump that they're using. A lot of the centripetal pumps and a few of the, not venturi. Yeah,
Diaphragm
you got it. Yeah, you got it. That's your diaphragm pumps. There we go. Diaphragm pumps will kill the organisms. So by the time your tea passes through that pump, you don't have any organisms left. So what's the point? You have to test your system and make sure that whatever pump you're using is not killing the organism. So again, you've got to have your microscope, or you have to have somebody local that will be able to help you with your doing your microscope work. So you want to find your closest soil food web consultant and get them to start helping you.
Any more questions? Yeah, you're here.
I got a question about the soil food web.
Yes, go ahead, you can ask it, and I'll repeat.
I'm just interested, I mean, outdoors makes sense, obviously, because when you transfer indoors.
Hi, I've got a question about the soil food web. When you transfer the idea of the soil food web back into an indoor environment, obviously you're going to be missing out on some of the last stages on your soil food web diagram, as far as birds, mammals, things like that. Obviously, we can't really add that in an indoor environment. Is there anything we can do as gardeners to try to mimic anything we're losing on the end of that?
Yeah, you would want to get beneficial insects into your greenhouse, so you get those higher level predators. Microarthropods, typically when you've got really good compost and it hasn't been turned that much. Typically with our thermal compost, we only turn them 3 times. We know that all of the pathogens and pests have been taken care of by heat, because we're very careful about the way we turn, making sure that all parts of that pile get into the hot middle. And so typically 21 days is all it takes to make good compost. Yes, we have to publish some of this data and work on the National Organic Standards Board to change their definition of what a good compost is. But when we're doing this work, we develop, there is a microarthropod community and therefore a macroarthropod community that's going to develop pretty rapidly in our greenhouse conditions. And using that really good compost, we're getting the microarthropods back in the yard systems, and that will hopefully attract the higher level predators. Not the birds, too large for a greenhouse system situation, but certainly the macroarthropods going to be coming along. It's when you start seeing spiders, big-eyed spiders, in all of the windowsills, on the floor, on the underside of your tables. Don't be killing them. Yes, if they're Lapidus spiders or gray recluses, you don't really want them hanging around, but the way you prevent those poisonous spiders from coming into your space is to make sure you've got the beneficial spiders. So wolf spiders, things like that, are not going to harm you. Harvesters are not going to harm you. So if you see a few of those in your greenhouse, just be happy they're there, because they're keeping all the rest of the food web below them happy. So did I answer that question? I had a hard time hearing the first
No,
part.
I think you got it. And I was just going to say, in a lot of the indoor systems, to be regenerative in them, we're going to have to do compost outside the building and creating this stuff and bringing it into the building. It just can't regenerate inside the warehouse, unfortunately. Some things will, but it's going to be part of the practice.
And also building birdhouses and habitat outside.
You say building birdhouses and habitat around your facility and building up the environment around your facility, pollinators, all the things. So we're going to get into a lot of this stuff, you guys. We all have experience. Most of the speakers, decades of experience growing indoors, outdoors, greenhouses. So let's get another question.
So you showed a diagram of the amounts of bacteria and fungi in certain systems by weight. I was wondering how we would go about measuring that in our own system.
We're going to do that using a microscope. So we're measuring the length and the width of the fungi. We're counting numbers of bacteria. We know the conversion factors to convert numbers or biomass into weight. So we get a biomass of the fungi, the biomass of the bacteria. Because you can't compare a single bacterium with a single fungal hypha. The largest organism on this planet is a single fungus, and right now the winner of the award for being the biggest organism on the planet belongs to a fungus that is growing in Olympic Peninsula in the old-growth cedar forest. And Paul Stamets is the person who monitors and keeps track of that. So that's a single fungal individual. Compare it with a single bacterium. You can't be comparing numbers. So you have to change, you have to measure biomass of the fungi and the biomass of the bacteria, which is weight. And so all of our comparisons are on a weight basis, comparing weight of the biomass of the fungi with the biomass of the bacteria.
And did you catch the ratios? We want to be, for cannabis, above 15 to 1, and 2 at 30 to 1. Yeah. Anybody else?
Sure. Can you just ask her the main biological differences between a compost extract and an actively buried compost?
Can you explain again the difference between a compost tea and a compost extract?
A compost extract is just the first part of making a compost tea. So you can take your compost and just use movement of water through that compost to extract the organisms off the surfaces of the compost and into the water. With a tea, we would then continue a 24-hour brew, and we would have put foods into the water before we add the compost. We would add additional foods to grow whatever organism we feel needs to be enhanced. So we would put in fungal foods if we need to enhance fungal growth during that 24-hour period. With bacteria, we might, under very rare conditions, want to improve the bacterial community, so we would put bacterial foods into the tea. It's just that most of the time, bacteria are not the missing organism, it's fungus or protozoa or nematodes that are the missing organisms. So we would put in an additional inoculum of protozoa during the tea brew to bring the protozoan numbers up. Or maybe we would put an inoculum of the nematodes extracted from really good, healthy compost or soil to increase the number of nematodes going out into your property.
What are the fungal foods and bacterial foods we can add to boost those at the beginning of the tea brew?
Yep, so fungal foods need to be fairly wide seed-in ratio materials. Oils, for example, are very good fungal foods, so fish hydrolysate, where they have not removed any of the oils, and you will probably have to be very persistent in talking to the person selling the fish hydrolysate to make certain that they haven't heated that fish hydrolysate and taken off the really expensive oils. Of course, all those oils are the things that are really the fungal foods. So you want all of those good oils in that fish hydrolysate. Humic acids are another good one. When you are making a compost, the actions of the bacteria and the fungi, protozoan nematodes are causing condensation of a lot of the organic material in that soluble organic material in your compost, and they're building those honey-colored compounds that we see in the aggregates. The really nice, rich, dark brown, 70% cocoa chocolate color that we see in the macro aggregates. So humic acids are being built by the microorganisms in your compost. If you take a container like a sieve, where you, for a pasta, you remove the water from your pasta, put cheesecloth on the inside of that sieve, and then put your compost. Passively run water through that compost. Don't shake, don't mix. Just let the water move through that layer of compost. Collect what comes out the bottom of that sieve. You can see the color of it. If it's just honey color, all you've got are the fulvic acids. If it's nice, rich, dark brown, 70% cocoa color, you've got the humic acids. And that is an excellent fungal food. And a lot of people will then say, well, but I'm extracting that humic acid from the compost. Now I'm going to add that humic acid to the compost. Yeah, you're increasing the concentration of that specific fungal food. You, of course, are going to use fresh compost, put into your tea brewer, and then you'll add the humic acid that you took from a different portion of compost. You can go ahead and put the solid material left in your sieve back into the compost pile, and the organisms start growing, and they just start making more humic acid for you. A lot of people will go out and buy commercially produced humic acid, but that is an extract, a chemical extract from soft brown coal, leonardite. And that process of extracting using really strong acids and really strong bases changes the composition of that humic acid, and it might take as long as 6 weeks to 6 months for the microorganisms to be able to utilize that chemically altered humic acid, humic and fulvic acid material. And of course, if you put that into a tea brewer where you're brewing for 24 hours, that commercially extracted leonardite-based humic acid is going to do nothing to improve the fungal component in your tea brew. So, food resources, other fungal food resources would be things like steel ground oats. Anything that they left the seed coat on when they ground it up is going to be good fungal foods. the germ inside of that seed is food to get those fungi to start growing really fast and start to utilize that seed coat material is what really selects for that fungal growth. So putting ground grain material, just making sure that it was steel ground or ground with the seed coat on it. They don't go and remove that seed coat first. When we're trying to really get this biology down into the root system, I mentioned injection, but another way to do it is to take your seed and spritz a good compost tea onto the surface of the seed, or immerse your seed into a compost tea for a few minutes. Pull your seed out, let them dry, and the organisms that get into the cracks and crevices, the seed coat of your actual seed material, will typically be able to hang out and wait somewhere around 2 to 3 weeks before you plant it. You're still going to get the benefit of those improved organisms on that seed. If when you're planting, like with your planter, if the first time going down into the soil, right after you open the furrow, then with the first time you deliver a compost extract to the soil, and then the next time drops your seed on top of that compost extract enhanced soil. That usually works really well at getting those root systems immediately colonized. And if you're using mycorrhizal fungi, you absolutely want to be doing the addition of the spores of the mycorrhizal fungi to the seed or to the soil right below the seed. If you're dealing with a seed that has been treated with fungicide or other pesticides, you have to be putting the extract into the ground before the seed goes on top of it. The pesticides they put onto that seed are going to kill anything that you apply to the surface of the seed, and it may well kill organisms in the 1 to 2 micrometer area below the seed, right around the seed. So you want that extract to soak into the ground, so when the roots germinate, when the seed germinates and the roots start growing down, they will get to that biologically enhanced soil.
We had a question on chat, and I think I can answer it about what microscopes you recommend. And I think the answer is go to the Soil Food Lab website, and there are 2, you talked about it earlier, but there are 2 microscopes that you recommend on that website. So another question, I think? In the beard, yeah, you got it, bro. No, you. Yeah.
Hi, thank you, Doctor. I had one question. At the start of your talk, you said something about anaerobic ferments being a pesticide and stripping the coating off of the fine roots. I just wanted some context on that versus historical treatment of anaerobic compost tea. That seems to be really successful, but I'm trying to rectify those 2. Thank you.
I need to have somebody repeat that, because you talk so fast and there's so much.
He was asking about using anaerobic teas, and earlier on in the talk, you talked about applying anaerobic teas as a pesticide. And coming from the experience of using anaerobic teas and seeing successful results in them, he's trying to make sense of this.
Yeah, the fermented teas grow anaerobic organisms, and those anaerobic organisms produce a number of toxic chemicals. They produce a number of organic acids that have a pH down around 2. So if you measure the pH on a fermented tea, typically the pH is going to be around 4 to 4.5. Not really beneficial to the growth of our plants, but this could be very instrumental in getting rid of diseases. So as long as you're not overwhelming the system with those fermented teas, you may be essentially putting on a pesticide. You kill some of the diseases that are in your dirt, and hopefully not to the point where you've absolutely put on some too toxic a level, but enough to kill the disease but not kill the plant. But really, we want to be dealing with that disease by having beneficial organisms doing all these other beneficial things for us. So you get much improved production. But it's a step that you can take, especially when you're first starting and you know you probably have some disease-causing organisms. Why not try to reduce those disease-causing organisms, but then come back in and inoculate with a really good set of biology? When we're looking at fermented materials, some of the other things that are produced only under anaerobic conditions, of course, are alcohols. And those are pretty good pesticides. Those kill a lot of the disease-causing organisms or inhibit their germination. So you can see a growth benefit to your plants, especially if you're growing in dirt, not in soil. I would probably encourage you not to use fermented feeds at all if you've gone through the work of getting a really good compost and you know you've got a really good set of beneficial organisms. Because with the fermented feed, you're going to be killing those organisms, the good along with the bad. So when we have fermented tea, we're going to be losing all of the soluble nitrogen in that tea because those organisms typically are going to be blowing off your nitrogen as ammonia, NH3. They're going to be blowing off your phosphorus as phosphine gas. They're going to be blowing off your sulfur as hydrogen sulfide. Exception to that fermentation problem is if you are loading your fermented tea up with a lot of lactobacillus. Lactobacillus is kind of a different story. They only produce organic acids. They don't volatilize your soluble inorganic nutrients, so not so much of a problem. So make sure with the fermented tea that you're actually adding lactobacillus and you're not allowing just any old anaerobic sets of organisms to grow in that fermented tea. So things like kombucha or kimchi, those are made with a specific inoculum of lactobacillus. So a lot less detrimental, other than pH is typically down around 4, 4.5, and you could be causing harm to your beneficial microorganisms if they're present. If they're not present, go for the fermented feeds. Yes, you probably would be a good idea to always do that if you're not dealing with soil. But as soon as you get the beneficial organisms and grow those beneficial organisms, stop with the fermented teas because they are detrimental to our good guys.
We can hit that more too tomorrow, Josh and Kelly, when they talk. I know they do a lot of ferments, so it would be cool to have them give their perspective. You, sir? Yeah.
Earlier you were talking about the ciliates that were like not beneficial organisms. They survived under 6 parts per million of oxygen, you said. I was wondering if there's a range of oxygen in soil that is, ideal for beneficial organisms, and if there's a practical way to test your dissolved oxygen in your soil, say throughout the season, to just ensure that you've got that ideal range for healthy microbiology to thrive.
Yeah, so the cutoff between aerobic and anaerobic is 6 parts per million oxygen. And I always like to measure the absolute concentration of either oxygen or carbon dioxide. They're kind of flip sides of the same coin. As oxygen is used up, CO2 is produced. So which one is easier to measure? CO2 is way easier to measure. You don't have as many problems with it. The probes for measuring CO2 in your soil or in your compost are much less expensive as compared to the oxygen probes. So I would encourage you to go after a CO2 probe. If you go to any one of the, like the bioags, the groups of companies that, Pike Labs, things like that, they have materials for making compost. We have CO2 probes for you. Just remember that the color indicator in a CO2 probe only contains about 100 measurements, and then you have to change the liquid inside of the probe, or it's just all white all the time, and you cannot detect CO2 or oxygen. So those are the simple ones. Usually you have a long pipe that comes with the probe. You push the pipe into the ground, and then you actually have to apply a suction to pull the soil atmosphere past your detector. So that's how compost or soils would be measured. In liquid, you would typically just take your sensor off and drop the liquid into your compost tea or your extract. You want an oxygen concentration, absolutely. You want it above 6 parts per million oxygen. Even better if you can get it up to 8. For most of our aerobic organisms, they're really happy in the range between 8 and 10 parts per million oxygen. Max oxygen concentration at sea level on a cold day at freezing temperature is 16 parts per million. So depending on your elevation and the temperature outside, you can get more oxygen into the air, but you know, we don't want to, we're not going to be growing good with the organisms at minus 32 degrees, which is where you can get higher levels of oxygen concentration.
So you said that those CO2 meters were a little bit more affordable and stuff. So did you have like an ideal level of CO2 that you're looking for when you're testing for CO2 levels?
Yep, so the flip side would be, typically with the CO2 probes, they're going to be looking at percent carbon dioxide acids. So anything below 3% is going to be very aerobic conditions. You can probably still be okay. You're in that range between 6 to 8 parts per million oxygen. You're looking at something like 3 to 6% CO2. Notice that I've switched units here. CO2 is usually measured as percent carbon dioxide or percent carbon dioxide plus other gases in the atmosphere. So you're again pulling a sample past your probe and you're measuring the percent of oxygen, CO2, and it's the percentage comes from the fact that the percent of CO2 plus oxygen, O2, should equal 100%. So as you use up your oxygen and it's converted to CO2, your CO2 value is going to rise. And you want less than 3% CO2 for really good growth of your aerobic organisms, but it can get up to maybe 6% and it'll still be okay. It's when you start approaching 8 to 9% CO2, you're going into, you're dropping into anaerobic conditions.
I'm just going to add, this is one reason that growing living soil in pots is very challenging. I won't say it's impossible because it's not, but it's very, very challenging to get the moisture level right, to keep the right guys playing. So recommendation is big beds if you can indoors. And Elaine will talk a bit about that. Go ahead, Elaine.
And don't overwater. We're just so tuned as human beings to, oh, the leaves are drooping a little bit, the plant looks like it's a little bit dry, so you add more water, and you go by the next time and wow, it's still looking a little bit droopy, I'll add more water. You're doing exactly the wrong thing, because those droopy leaves, that unhappy attitude of the plant is probably telling you that the water is waterlogged, the soil is waterlogged. It's going anaerobic. Well, oxygen does not move through water very rapidly at all. So you have to not let that instinct of, oh, it's not looking real happy, I'll add more water. Don't do it.
Okay, one more, sir.
Thank you. For soil compaction that's 6 inches or a foot down, it sounded like the best thing to do was to till and then to add compost tea to add biology back to the soil. For your example that you showed where the soil compaction level was at 6 or 8 feet down, what can you do to eliminate that compaction and to get life back in the soil?
Yeah, the easiest thing to do for a very deep compaction layer is to get an injector. And so you want an injector rod that can go down a good 4 feet, especially if you've already gotten good structure in the upper layers of the soil. You know, you have no compaction layer in that top 4 to 6 feet. Then you can get a really long metal pipe and just screw it into your injector on the end of that gun. And then you push that all the way into the soil. You're going to want to push down and hopefully through the compaction layer. So you're injecting that really good biology through that whole compaction layer up all the way through your soil. Hopefully then, by the time your roots actually get down to that compaction layer at 6 feet or deeper, the biology will have had the chance to break that all up so that your roots will be able to grow down, possibly can.
He's asking how to inject. Do we inject every 3 feet, every 2 feet, every 6 feet? Like how are we applying in this scenario? Is that correct?
Yeah, what we've seen is that if you're injecting into the soil pretty much at the maximum pressure for releasing, you probably can go every 10 feet because the biology is being pushed out sideways, and under that amount of pressure, the biology is going to be moving into all the cracks and crevices that water can open up for it in all directions as well as down. And so what we see with this injection, the diameter goes out about 5 feet. And so if you put another injection hole 10 feet away, then they're going to overlap the amount of distance that water is going to move sideways. It's going to overlap with the next injection that you do. And then of course the organisms keep growing through the whole remaining part of growing season. So you're hopefully also going to be coming in with a compost tea spray to cover the above ground part of the plant. And when you're applying a spray to the foliage, you are also going to be dripping, misting your soil surface as well. And then hopefully you'll be watering or it will rain. So that biology will be able to move down deeper into the soil.
And this is actually stuff that is Layton's specialty. He's designing really brilliant growers and pliers and shelf-stable compost. So I'm going to let him talk a little bit more about that. But Elaine, I just wanted to say I didn't get to introduce you proper. Thank you so much for all the work you've done over all the years. You've definitely changed my life. And the fact is, it ripples. You know, I'm able to grow healthier plants. We're on stage talking about this. So really, truly, thank you for the work you've done.